Hello everyone, I'm Anas Al Ubaidi, and this is the 10th lecture or 10th session within Chapter 20 of the Advanced Medical Science Liaison and Medical Monitor Certification course program by CCRPS. And the chapter is about the specialized monitoring techniques, while the topic for the session is about the monitoring of hematology and psychiatry trials. Introduction. Clinical trials in hematology and psychiatry are among the most intricate, requiring tailored monitoring systems to accommodate the challenges posed by such vastly different yet equally complex disciplines. Hematologic trials often center around life-threatening diseases such as leukemia, lymphoma, or blood clotting disorders and may involve high-risk interventions such as chemotherapy or stem cell transplantation. Meanwhile, on the other hand, psychiatry trials evaluate treatments for disorders like depression, schizophrenia, and bipolar disorder, which require monitoring of subjective outcomes and unique adverse event profiles, including cognitive and behavioral changes. The key points for the topic will start with the monitoring techniques in hematology trials. Hematology trials often involve high-risk treatments that require close and continuous monitoring of patient biometrics, response rates, and adverse event profiles. The core monitoring areas, despite of the order, will start with patient safety monitoring, vigilant tracking of hematologic parameters like hemoglobin, white blood cell counts, and platelets is essential to identify and address emerging safety concerns, as for example, monitoring platelet values and clotting times in trials for novel anticoagulants. Tools centralized data systems can alert trial staff to critical drops in cell counts, enabling immediate intervention. Another core monitoring area, protocol adherence. Precise adherence to treatment protocols, including chemotherapy regimens and transfusion schedules, is critical for trial success, as, for example, ensuring a precise dosage schedule during stem cell transplantation for acute myeloid leukemia (AML) for short patients. And before we proceed, we have here a summarized diagram on hematology trial monitoring, uh, summarizing patient biometrics, adverse event profiles, and response rates. So, continuing with the core monitoring areas, adverse event documentation, AE documentation, capturing treatment related hematologic toxicities such as anemia, infections, and/or bleeding events ensures compliance with regulatory standards. As for example, reporting pancytopenia in a myelodysplastic syndrome trial via MidWatch. And MidWatch is one example of software where to report AE to or document the AE to. Biological sample handling, another core monitoring areas, one of the core monitoring areas, biological sample handling. So proper storage and timely analysis of blood, bone marrow, or genomic samples ensure data accuracy and reproducibility. As for example, cryopreservation of bone marrow aspirates for molecular analysis in myeloma trials. Tracking, another core monitoring area, tracking clinical outcomes, hematologic response criteria such as complete remission (CR) or progression-free survival must remain well documented. As for example, utilizing the International Working Group (IWG) criteria to track remission in AML, acute leukemia trials. The key takeaway. Monitoring in hematology trials emphasizes rigorous adherence to safety and protocol specifications to minimize risks and ensure participant protection or patient protection. The monitoring techniques in psychiatric trials, as psychiatric trials require the monitoring of subjective outcomes, behavioral changes, and medication and adherence to ensure reliability data and participant well-being. So the core monitoring areas: symptom tracking with validated skills. Regular use of psychiatric assessment tools provide a quantitative basis for determining treatment efficacy, as for example, administering the Hamilton Depression Rating Scale HAM-D weekly during an antidepressant trial. Cognitive and behavioral assessments, another core monitoring area, monitoring subtle changes in attention, memory, and behavioral protects participant safety and informs intervention strategies, as for example, using the positive and negative syndrome scale PANSS, PANSS for evaluating schizophrenia symptoms. Before we proceed, we have here a summarized diagram showing the monitoring in psychiatric trials, as we have medication adherence, subjective outcomes, and behavioral changes. So, continuing with car monitoring areas, we have medication adherence. As adherence monitoring prevents confounding data that could result from partial or non-use of study medications. As for example, utilizing smart pill or pill bottles that track when medication is accessed. Adverse event detection, another core monitoring area. Psychiatric trials necessitate particular vigilance for psychiatric AEs, adverse events, such as suicidal ideation or agitation. As for example, recording trends of self-reported paranoia during antipsychotic trials. Patient safety measures, another core monitoring area. Regular assessments for warning signs like aggression or hallucinations help prevent crisis, as for example, immediate notifications to clinicians about severe hallucinations in schizophrenia participants. The key takeaway: robust monitoring in psychiatry trials ensures safety through consistent use of validated psychiatric measures and vigilant tracking of adverse psychiatric symptoms. Then we have data analysis in hematology and psychiatry trials, as effective data analysis is central to risk management and generating actionable trial outcomes in both domains. Data analysis techniques: we have the risk-based monitoring, RBM, focusing resources on high-risk elements within hematology studies with complex biological markers, or psychiatric trials with subjective outcomes, so forecasting risks. Example: RBM, risk-based monitoring systems flag abnormalities in white blood cells account during thrombocytopenia, in white blood accounts or white blood cells accounts during thrombocytopenia interventions. Then we have predictive analytics, as another data analysis technique. Historical trial data is leveraged to anticipate adverse events, AEs, or patient outcomes, as for example, predicting relapse rates in lymphoma patients under experimental immunotherapies. And here is a summarized diagram on the data analysis, as here we have divided into four areas: high impact, high complexity, low impact, low complexity, and here we have descriptive statistics, advanced statistical modeling, machine learning algorithms, and basic data entry. So continuing on data analysis techniques, we have machine learning ML applications. Advanced algorithms detect meaningful patterns in participant responses or AE risks, as for example, identifying patient profiles in schizophrenia trials prone to non-compliance. Centralized data review, another data analysis technique. Consistent consolidated review across sites ensures high data integrity. Example: real-time analysis of missing behavioral scores in multi-center psychiatry trials. The key takeaway: leveraging advanced analytic techniques reduces trial inefficiencies, improves decision-making, and boosts safety outcomes. Then we have challenges, as any challenges we face or we might face during any implementation in clinical trial, challenges and solutions or resolutions. Challenges: complex trial designs. Hematology trials require integration of genomic and clinical data, while psychiatry trials need subjective outcomes tied to behavioral metrics. Solution: standardize workflows using cloud-based platforms. Poor adherence in psychiatry trials. Stigma or cognitive deficits reduce compliance rates. Resolution or solution for that: implement reminders, telehealth check-ins, and gamified adherence tools. And then we have, as another challenge, data overload. Both types of trials generate significant amounts of data from wearable imaging and questionnaires. Solution resolution for that: automated aggregation and analysis platforms simplify data management. And here is a summarized nice diagram on the challenges and resolutions as high data management needs, low complexity, high complexity here, and low data management needs. As here we have standardized cloud workflows, automated data platforms, telehealth check-ins, and gamified adherence tools. And the key takeaway here: addressing these challenges with innovative tools improves trial execution and ensures meaningful results. The recent advancements in monitoring, as we have advanced technologies, are redefining trial monitoring by enhancing data precision and patient engagement. So, um, one of the advancements, we have wearable sensors measure vitals or behaviors in real time. Is, for example, Fitbit as devices, as one example of software, track sleep changes in psychiatry trials. Another recent advancement, also classified as recent, genomic profiling guides precision medicine in hematology trials. Example: targeted mutation analysis predicting response in leukemia therapies. Another advancement: telemedicine integration expands reach across underserved populations. Here's a summarized diagram of a brain shaped, as here we have it under summarizing or showing the enhancing trial monitoring, as we have data precision and we have patient engagement. Example: psychiatric symptom monitoring through virtual integrations in rural areas. Another advancement, we have real-time monitoring dashboards offer centralized views for anomaly detection. Is, for example, global overview of platelet trends during chemotherapy trials. The key takeaway: these advancements amplify the ability of physician monitors or advanced physician medical monitors or the medical monitors, MMs, to achieve protocol adherence and participant safety at scale. By mastering these strategies and leveraging innovations, advanced physician medical monitors, MMs, can confidently oversee hematology and psychiatry trials, ensuring robust, ethical, and impactful outcomes. Next, we have tables to summarize what we have covered through this lecture. We have table on hematology monitoring: focus area, task, example, outcome, challenges. Focus areas: adverse event monitoring, protocol adherence, biological sample handling, clinical outcome tracking, and safety markers. Another table on psychiatry monitoring techniques: we have method, focus, application, benefits, limitations. As methods, we have psychiatric symptom scales, behavioral observation, digital pill tracking, neuropsychological tests, and patient safety alerts. Another table on data analysis and trials: analytical method, purpose, example, advantages, and then challenges. Analytical methods: we have risk-based monitoring, RBM, machine learning algorithms, signal detection analytics, predictive analytics, and centralized review systems. Another table on benefits of effective monitoring: as benefit, outcome, example, reason for benefit, and then challenges addressed. As benefits, we have enhanced patient safety, data consistency, resource optimization, accelerated decision making, regulatory compliance. Table on emerging innovations, innovation, functionality, example, advantages, and then limitations. As innovations, we have wearable technology, genomic profiling, telemedicine platforms, real time cloud analytics, AI, artificial intelligence driven adherence alerts. Then we have application table as scenarios for each scenario, key principle applied, key actions, timeline, what to watch out for. Scenarios we have leukemia trial monitoring chemotherapy side effects, schizophrenia trial evaluating antipsychotic efficacy, lymphoma trial with multi-site patient recruitment, bipolar disorder trial tracking treatment adherence, stem cell transplant protocol in myeloma patients, depression trial using wearable health devices, anticoagulant study after major surgery, hematology pediatric leukemia genomic study exploring relapse risk, suicide risk assessment in an anxiety treatment trials, remote psychiatric trial in underserved populations. Next, we will be revising what we have covered through this lecture via some multiple choice questions or MCQs. As question 1, why is monitoring patient safety critical in hematology trials? Is it A, to ensure proper storage of medication bottles? B, to continuously track hematologic markers like hemoglobin and platelets? C, to avoid conducting site visits? Or D, to reduce participant enrollment time? So, why is monitoring patient safety critical in hematology trials? Doesn't make sure C, to avoid conducting site visits. Why would be that? And does it make sense to reduce participant enrollment time? Um, A, to ensure proper storage of medication bottles is weak as to be considered as a reason. So the most appropriate answer would be B, to continuously track hematologic markers like hemoglobin and platelets. As hematology trials often involve monitoring vital blood components to promptly identify adverse events AEs such as anemia or thrombocytopenia, ensuring the safety of participants. Question 2. What is a key method for monitoring adherence in psychiatric trials? Is it A, conducting biweekly on-site diagnostic reviews, B, using pill counts, interviews, or digital applications, or C, relying on participants' memory for medication reporting, or D, ignoring adherence issues during early trial phases? So does it make sense as a key method for monitoring adherence in psychiatric trials? Does it make sense relying on patients' or participants' memory? And does not make sense ignoring adherence issues? Um, Conducting A, as conducted biweekly on-site diagnostic reviews, is very weak, as the stronger the answer and the most appropriate answer would be B, as using pill counts, interviews, or digital applications. So monitoring adherence through structured methods like pill counts or digital tools ensures participants follow their prescribed treatments, which is vital in psychiatric trials where non-compliance is common. Question 3. Which of the following challenges can be addressed during cloud-based analytics in clinical trials? A. Increasing participant recruitment. B. Managing large sets for multiple sites. From multiple sites, avoiding regulatory compliance requirements, or D. Ensuring exact biological sampling locations. So B would be the reasonable answer, as managing large data sets. That's why cloud-based analytics, cloud as large data, dealing with large data sets from multiple sites. So B is the answer. As a cloud-based analytics, centralize and streamline data from various trial sites, helping monitor large-scale studies efficiently and with greater accuracy. Question 4. How can signal detection improve monitoring in psychiatric trials or psychiatry trials? A, by identifying patterns in adverse events, AEs like rising suicidal ideation rates. B, by reducing reliance on participant-reported outcomes. C, by simplifying adaptive design methodologies. Or D, by increasing the time taken to analyze safety signals. So most accurate answer would be B, by identifying patterns in adverse events like rising suicidal ideation rates. As signal detection tools analyze trends within trial data to flag early safety concerns, allowing for timely adjustments in trial protocols, especially in psychiatry trials. Question 5. What is a significant application of predictive analytics in hematology trials? A. Identifying manual errors in symptom reporting. B. Reducing the need for adverse event documentation altogether. C. Predicting relapse risks or progression free survival trends. Or D. Eliminating the use of RBM risk based monitoring. So, doesn't make sense to eliminate the use of RBM, nor identifying manual errors. No. Reducing the need for adverse event documentation altogether also doesn't make sense. So what's left? The most reasonable answer would, or and the most correct, accurate answer would be C, as significant application of predictive analytics would be predicting relapse risks or progression-free survival trends. As so, C is the right answer. As predictive analytics uses a historical and real-time data to forecast critical outcomes like survival trends in patients, helping refine treatments in hematology trials. Here we come to end of this lecture. Thank you very much and goodbye.